Hello, and a very warm welcome to everyone who's joined us today. My name's John Norman. I'm Head of Marketing at Optibrium and moderator for today's webinar. As always, it's great to see so many people signing up for our webinars. And today we will be discussing imputation of sensory properties using deep learning. This is a case study from International Flavors and Fragrances. And for today's presentation, I'm joined by Samar Mahmood, who is Senior Scientist at Optibrium. Our host today is Tamsin Mansley, Head of North American Operations. And I'm also delighted to welcome our guest, who is Dmitriy Chekmarev, a Senior Research Investigator at IFF.
Thank you, John. As John said, my name's Tamsin Mansley, and I'm the Head of North American Operations at Optibrium. And it's my very great pleasure to welcome our guest speaker today, Dmitriy Chekmarev. Now, for Optibrium, this was an especially challenging project that we're going to be talking about today, where we were interested in investigating the benefits of working with especially noisy and subjective data, especially sparse data as well. The majority of our previous projects have focused on pharma data, but this case study shows that the imputation approaches that are used can really be generalized beyond pharma. Dmitry, you have experience working both in pharma and in flavors and fragrances. Could you tell us about some of the challenges that you face in flavors and fragrances at IFF that are perhaps different to those that might be experienced in drug discovery?
The main difference are in the type of endpoints that we consider, because in a sense and food science, we totally rely on human perception. We rely on sensory experiments and how human beings perceive different odors or tastes. That actually makes it more subjective because it has a higher level of noise in this data, but also it's actually the data that's more difficult to collect because to run these experiments, you need to recruit, train and assemble panels of human testers. And you can imagine that especially right now in the times of COVID, it's very difficult to run these experiments. So we became interested in Optibrium deep learning technology because it offers an possibility or opportunity to retrospectively fill the data set that we collected over many years and which still have a lot of missing data set of sensory data with reliable prediction. And I will speak, and Samar will actually speak about this more.
Thank you. And while we're talking today specifically about odor detection, would it be right to say that the principles apply to any sensory data?
Oh, absolutely, yes. We can actually explore the similar ideas and apply it to test sensation. And in that respect, actually, probably will bring it closer to pharma because test modulators, some of them are very similar to drug-like molecules. So yes, absolutely.
That's great, thank you. And so let's dive into the main part of today's presentation. As you can see, Dmitry is going to start with an introduction to sensory properties and the data used in this project. Then he's going to hand over to Samar, who will present the Alchemite method that we used for deep learning imputation and the results that we obtained. We'll all come together for a discussion of the conclusions, and then as we go through the presentation, as John mentioned, please feel free to type your questions into the questions panels, and we will address those at the end. I'm also very excited to announce that this work has just been accepted for publication in JCAMD, and we've put a link to a preprint of the paper at the bottom of this slide. So do feel free to jot that down if you'd like to be able to go and read a little bit more detail. So without further ado, Dmitry, I'll hand the presentation over to you.
So let me start with a quick introduction to IFF. IFF, International Flavors and Fragrances, is a company with more than 100 years of history and is one of the global leaders in scent, food, beverage, and health sciences. We have 4 areas of concentration for which we create products and developed creative solutions that are nourish, scent, health and biosciences, and more recently pharma. So today we'll focus on one scent-related application, and specifically we ask the question of prediction of 2 key olfactive properties, the odor intensity and the odor detection threshold for fragrance ingredients. Both of these properties, together with the odor character itself and the number of physicochemical properties, determine how well this particular ingredient, fragrance ingredients, will perform in different fragrance applications. Odor intensity dose response curve is a curve which can be traced through or fit to a number of evaluation of odor intensity performed by a panel of trained panelists at a series of different concentrations or dilutions. As shown on this right plot, you see that this is the bold curve. And this is shown for Veromos. This is a molecule that's known for its mossy, woody odor. And panelists I asked to mark the perceived intensity on the labeled magnitude scale, which is a quasi-logarithmic scale of intensities relative to some standard benchmarks, and which has this kind of verbal anchors to show the intensity, possible intensity ranges. So the knowledge of this relationship helps perfumers to correctly dose different ingredients in a complementary fashion in different formulations and applications. The second quantity is the odor detection threshold. This is the lowest concentration that is detectable 50% time by the panelists. It's marked by this dashed line on this plot. And also this helps prioritize different ingredients, different high-value ingredients for different formulations. Both of these quantities are results of human evaluation, and as such, they carry considerable amount of noise because they're being subjective by its nature. And also there is a certain degree of inter-subject variability due to natural difference of odor perception, of perception of different odors among human population. And finally, these experiments are expensive and time consuming. And as you perform these experiments over time, you started accumulating a number of gaps, a number of missing points, which due to undone experiments for a variety of reasons. And it would be very beneficial to have a model which can actually use the available chemical information and some available experimental data and can fill these missing gaps reliably with some prediction based on this chemical structure and available little experimental data. So this is exactly what got us interested in Alchemite deep learning approach, because this is the approach which can use to predict several properties for multiple compounds from a very sparse data. And exactly the questions that we wanted to ask, we want to see, to assess the ability of Alchemite to impute missing structure property data, both sensory and physicochemical, in the station when you have both chemical structure available and also some limited experimental data to support this prediction. We also wanted to see how Alchemite performs when you don't have experimental data, and you rely only on available chemical structure. And naturally, we would like to compare Alchemite with the standards, or I will call it conventional machine learning methods, which are developed to predict such properties. Let me then quickly guide you through the data set which was used for this study. This data set included 1,094 molecules drawn from IFF internal proprietary catalog, and 7 properties were actually selected for modeling: 2 physicochemical properties, vapor pressure and water solubility, and 5 sensory properties, odor detection threshold, ODT, and 4 points along the odor intensity dose response curve. This from I1 to I4. All these properties together, they actually define the performance of each ingredient in various applications. As you can see in this table, they have the varying degree of sparsity. And our specific interest was attached to prediction of odor detection threshold because first it has almost like half of the data missing. Then it's the least correlated property among this set of 7 important properties. And actually, it's notoriously difficult to model it by itself. This data set can be considered as a representative data set for a typical fragrance catalog because it includes compounds from all major chemical classes, as shown on this plot, and also across all major odor categories, as shown on this plot. So the data set was split into the training and test subsets, roughly the proportion of 85 to 15%. The experimental part was blinded, and the training portion, along with the chemical structure, was sent to Optibrium for model building and validation. And when the models were ready, we provided Optibrium with the test set for independent evaluation. So, because we're mostly interested in filling in this missing data points in our database, we decided to select the compounds in the test set such that they will actually maintain the observed distribution in both, maintain the diversity in chemical space, as shown in these 2 plots, acceptable distribution against all major odor categories, and also show decent distributions, consistent distribution between the training and test sets for all the properties of interest that we would like to model in this study. And with that, I pass control to Samar, who will describe the algorithm and the results of the study.
Thank you, Dmitry, and welcome everyone, and thanks, Tamsin, for the introduction. I will be describing the application of the deep learning method on the sensory data that Dmitry described, but before that, I'm going to describe the method that we used for deep learning imputation. But first, let's make a distinction between the prediction and imputation. So we are all mostly familiar with the term prediction, which is the process of predicting one or more property for a compound using descriptors as input. So if we imagine that we have compounds that are described by descriptors, we can build a machine learning model or a QSAR model, let's say a random forest in this case, to predict one or more property. And it's essential here to distinguish that the input descriptors that are used in the QSAR model has to be a complete matrix where all compounds have 100% of the descriptors calculated, whereas the imputation is the process of filling the gaps in a sparse experimental data. So the starting point is a sparse experimental data matrix that these sparse data are used to impute the missing values in the sparse matrix, and in this way, the imputed values are generated for the gaps in the experimental data. And so the deep learning imputation method that we've used is called Alchemide, which is developed by our partners Intelligence, which is a spin out from the University of Cambridge. And the key advantage of the Alchemide method that it's used, that it learns the relationships and the associations between experimental endpoints, in this case, the assays, in addition to the structure-activity relationship. So it takes the molecular descriptors as input, in addition to the sparse experimental data, in a way to learn both the structure-activity relationship and the relationship between experimental endpoints to generate the predictions. And in this way, it can make better predictions than the conventional QSAR models because it leverages the relationship between experimental endpoints. And in this way, it can fill in the gaps in the sparse experimental data for tested compounds. However, the model can also generate predictions for virtual compounds or compounds that have not been tested using molecular descriptors only as input. And in this way, it can generate accurate predictions, which enables to target and identify high quality compounds. And a second key advantage of the Alchemite method that it generates an uncertainty estimate for each prediction. And that's that method it does not only generate a single prediction for each compound assay point, but rather a probability distribution. And so we can use that distribution to determine how confident the model is about that prediction. And so in cases where the prediction is wide, it means that or the distribution is wide, it means that the model is not very confident about the predictions. However, in cases where this distribution is sharp, it reflects a prediction that is generated with high confidence. And in this way, we can highlight the most confident predictions that reflects the most accurate results. And in this way, we can use the uncertainty, the estimated uncertainty, to target the high quality compounds. And in cases where the model is less certain about the predictions, we can use that information to actually prioritize experimental resources. And so in this collaboration, we've applied the Alchemite deep learning imputation method on the sensory data that Dmitriy explained. And the application scenarios for the Alchemite method it can be applied in two ways. And it's dependent on the availability of the experimental data for compounds in interest. So one application scenario is the imputation model, which generate predictions for compounds that have been sensitized and tested using the sparse experimental data and molecular descriptors as input. Whereas a virtual model is used in the cases where we want to generate predictions for compounds that have not been tested or sensitized, so we don't have any experimental data for these compounds, and so the models are built to expect only molecular descriptors as input. And so in order to use these models, both the imputation and virtual model, to generate predictions, to fill in the gaps in the data, or to generate predictions for virtual compounds, we need first to validate these models. So the process to validate first the imputation model, we start with the experimental matrix that includes sparse data. So we have a matrix of sparse experimental data where we have, in this case, compounds in rows and assays in columns. We split the data by compounds into training and test, and then the training set is used to build the model, where we calculate the descriptors for each compound in the training, and then the model would learn both the structure-activity relationships and the interassay correlations between experimental endpoints. The model is optimized in a cross-validation procedure. Then the optimized model is applied to the test set in order to generate predictions for all of the measured values. So for example, if we take this red square as an example, the model would use the vector of descriptors for that compound and any available experimental data for that compound. And so the model would generate an imputed value, which we can take back and compare it with the actual measured value. And in this way, we can assess the performance of the model. In a similar way, the virtual model is built and optimized using the training set, where again, it also it learns the structure-activity relationship and the interassay correlations between the assay in the data, and optimized through 5-4 cross-validation. However, the model is optimized in a way to expect only molecular descriptors at test time, so no experimental data are used as input. So it's applied on a test set that has includes only molecular descriptors as input, and so it generates predictions for these compounds where no experimental data are used. And for the then we take the predicted values for test compounds and compare it to available measured data points. And this way we can validate the virtual model. And the way we assess results of the Alchemite models is by using two ways. So one way is looking at the performance of the model overall, and we can calculate the coefficient of determination as a metric for accuracy for each individual endpoint. And the coefficient of determination, the R squared, is a metric that ranges between -infinity and 1, where value of 1 means 100% accuracy, and 0 means similar to random, and a minus value or negative value means worse than random. And so after we calculate the coefficient of determination for each endpoint on the test set, we can rank the endpoints based on accuracy, and we can generate a performance profile by plotting the endpoints ordered by accuracy in the x-axis and the coefficient of determination R squared in the y-axis. So we can generate a profile similar to this one, where the higher the performance is represented roughly by the area under the curve, or in other terms, if the line goes towards the top right. And an estimate for the overall performance is looking at the median R squared across all endpoints. We can also examine the performance of the model in individual endpoints using the error as metric, the root mean square error, the RMSE. And so we are not only examining the performance of the model on the endpoint overall, but we can also assess the uncertainty estimates by the model on individual endpoints to see how whether the most confident results reflect the most accurate predictions. And we can do that by ranking the tests, the predictions by accuracy, by confidence in the x-axis versus the RMSE in the y-axis. And so for 100% of the predictions, we would expect to see the highest error or the least accuracy. But as we go towards the left, where we remove predictions with the lowest confidence or with the highest uncertainty, and focus on the subset of predictions that have the highest confidence, we see that the error drops, which reflects an increase in the accuracy. And in this example here, we see a good correlation or strong correlation between the estimated uncertainty and the actual accuracy. And so this is quite important to examine this, because we can use these estimated uncertainties in cases where the model might not generate a great model for an endpoint overall, but we can still identify the most accurate results using the estimated uncertainties and by focusing on the most confident results. So upon applying the Alchemite model in the training set, we've generated within the training set on a further split for a validation, internal validation set. And we compared the Alchemite imputation model with QSAR and Chemprop models. So we are comparing here 5 models: the Alchemite imputation, the Alchemite virtual, 4 QSAR models, and the Chemprop deep learning method. So the Chemprop in this case is a graph convolution neural network method that learns directly from the graph structure of the compounds, and it constructs its own molecular encodings that represent the features, the vector features of each compound, using the convolution centers on bonds. And so for comparison, we've also built Chemprop models using the same descriptors we used for the other models. So here we make a distinction between the Chemprop graph and the Chemprop descriptors. So the plot here represents in the x-axis the different endpoints used in this analysis. So the vapor pressure, and the water solubility, the first two, and then 5 sensory properties, including the ODT and 4 odor intensity endpoints. And what is apparent here that the outcome at imputation, which is represented here by the blue line, has higher R squared over the sensory properties, the ODT, and the 4 odor intensity endpoints, in comparison to the other virtual models or molecular descriptors-based models, which shows how the benefit of using the imputation model over other models. And the reason for that, because it's the imputation model leverages the inter-assay or inter-property correlations in a way that adds extra information on top of the structural information, even if the data is sparse and noisy. We see that also that all models perform similarly over physicochemical properties, the vapor pressure and the water solubility, because these properties are captured very well using by the structural information. And for the sensory properties, especially the ODT, we see that the QSAR and the ChemProp models struggled most in predicting this property, where we see a great advantage by the imputation model, achieving an R squared of 0.6 versus lower than 0.4 R squared using other methods. The outcome at virtual also we can also observe a small advantage over the other QSAR and ChemProp methods, because although it's not using the actual experimental data as input, it still learns the relationships between the different properties, and so it can provide benefit over the conventional QSAR methods. And so then we applied the optimized Alkamite method on the independent test set. We see similar patterns, again, where the imputation method outperforms the other models over the sensory properties, especially for the odor intensity endpoints, and that confirms the significant benefit by the Alkamite imputation, because it leverages the inter-property correlations. We also observe that across the different sensory properties, the virtual models again outperform the QSAR methods, because it learns these it makes the most out of the inter-property correlations, even though it's not getting any experimental data as input, which shows again the benefit of Alkamite method over other QSAR method. It's interesting also to see that the QSAR method struggled most with the odor intensity I1, which is perhaps because of the high variability between the test subjects to determine or to detect the odor at the lowest concentration. And so when such subjective or high variable endpoint can be hardly detected or hardly captured by conventional QSAR method, the Alkamite method have shown substantial benefit in predicting such endpoints. It's also worth noting that how the ChemProp graph-based method had deteriorated in performance in comparison to the validation set, which reflects that the representation learned by the ChemProp method from the training set did not capture the structure activity relationship of test compounds. So it was not able to extrapolate to the chemical space of test compounds. But overall, the most significant results here is how the data have benefited from the Alkamite imputation model, because of leveraging these inter-property correlations, even if the data is subjective, noisy, and sparse. So let's focus on the oddity endpoints, since it had the highest interest by the IFF, and here we are comparing in the scatter plot the predicted values for the oddity versus the observed values, and here we're comparing the outcome imputation with the best QSAR model, where we see a substantial improvement in the R-squared, and also we see how the points of the predicted value by the best QSAR model are scattered in comparison to the points predicted by the imputation by the alchemite imputation method, and that illustrate the higher value or the higher R-squared estimate. And so we can also look at the estimated uncertainty by the alchemite imputation model on the oddity endpoint. And again, here we are looking at the observed oddity values versus the predicted values. However, in this case, we are adding an error bar for each point which reflects the uncertainty, the estimated uncertainty by the alchemite imputation model. And so for in this case, for 100% of the prediction, we see the highest error as in the plot on the left. So the RMSE is around 0.9 for 100% of the points. But we can leverage these uncertainty estimates and remove these points which has the highest uncertainty and the largest error bars. So if we focus on the most confident 50% of predictions, we see how the points become closer to the identity line and the overall error drops significantly. And we can even further focus on the top confident 20% of the data, and we see that the error drops further. The points becomes closer to the identity line, and so we here observe the strong correlation between the estimated uncertainties or the model confidence here represented as error bars and the observed accuracy. And this is quite an important finding, because it means that we can reliably use these estimated errors to focus on the most accurate predictions. We also examined how the model performs with regards to the chemical space. So we've compared the alchemite imputation model with the best QSAR model for the ODT with regards to the top 20% of the test compounds that are least similar to training tests. So we've seen that for the compounds that within the test set that are least similar to training, they show the highest error within the QSAR model. So if we looked at the absolute difference between the measured and the predicted value, the variability in the predictions between the variability between the predicted and the measured value, we see that within the least similar compounds, it's highest among the best QSAR model. In comparison to the alchemite imputation model, we will see a small error. And what this means is that the alchemite imputation method extrapolate better in the chemical space than the QSAR model. And the reason for that, because it uses both the structure activity relationship captured by the structural information in addition to the property space. So it uses both the information within the chemical space and the property space. Even if the data is sparse and noisy, it can make significant improvement in the accuracy of predictions. What was also interesting in this data that we've observed several activity cliffs. And activity cliffs are compounds that are characterized by high similarity in structure, but large changes in the property values. So here there is an example of 3 test compounds that are very similar to the nearest neighbor in the training set. Actually, the difference between these test compounds and the compounds in training is a methyl group. And what we can see is that for the oddity values for the for these test compounds are significantly different from the nearest neighbor in the training set. So if we looked at how these compounds have been predicted by the best QSAR model, we see that the predicted value by the best QSAR model is far from the actual value, and it's closer actually to the nearest neighbor of the in the training set for the 3 compounds. Whereas for if we compared the predicted value by the Alkamite imputation model to the actual observed ODT value of the test compounds, they are quite similar. So, and which means that there is a much smaller error in the predicted value by the imputation model in comparison to the best QSAR model for these activity cliffs. So these examples here show how the QSAR model often fail to identify the large changes in property with such small changes in compound structure. But on the other hand, the Alkamite method succeeds because the prediction is based both on the structure information and the sparse experimental data were available for the related properties. And so we've seen how the Alkamite deep learning methods have been applied to sensory property data, which is characterized by being noisy and having the inter-individual variability and subjectivity between the test subjects. And in this way, it presents a challenge for predictive modeling. However, we've seen how the Alkamite imputation model offers a significant advantage over the conventional QSAR and the multi-target graph convolution neural networks. And that is despite the sparsity in the experimental data, the Alkamite can still extract the additional information from the data, even though it's sparse and noisy. We've also seen that how it can show advantage in extrapolation in the chemical space and also detecting the activity cliffs. One of the significant findings is that the uncertainty estimate by the Alkamite model can be reliably used to identify the most accurate predictions. And in this way, it can be used to make decisions. We can make decisions based on the most confident results and also avoid missing opportunities by rejecting compounds based on inaccurate data. And so these summarize this collaboration that we have with the IFF and present the key findings. And I would encourage you to look in the paper that have just been accepted, where you can download it from the link below.
Thank you very much, Samar. I think this was a particularly challenging project, and your presentation very nicely illustrates the benefits of applying the imputation approach to particularly sparse and subjective sensory data. I was especially struck by the benefits of imputation to predicting the odor intensity at low concentrations, where you mentioned there might be a lot of variability between the perception by the different panelists. Dmitry, do you have any comments about the results from the project and the value that this has brought to IFF?
I just want to add that we were very encouraged to see such a big improvement in estimation and prediction accuracy for properties such as odor detection threshold, just by inclusion of a small amount of experimental data that is not even like highly correlated with this property. So this is definitely a right step in the right direction toward bridging the gap between the accuracy of the best available model that we have right now to predict sensory properties and this potential upper practical limits on this predictability due to the inherent noise that's in this data. I also want to see, want to emphasize that using this uncertainty estimates from Alkamite model, we can actually look back at our data set and mark those compounds that probably need to be retested, and also check those compounds that may actually present some kind of new information to us. That might be because large uncertainty, because this is the new chemical series. So we can, and it's definitely worth exploring it further.
That's a great point. Yes, a couple of the big advantages of this approach are highlighting false positives or false negatives, and also some of those unexpected results like the activity cliffs that Samar mentioned. Another thing that occurred to me is that in flavors and fragrances, by using the human panelists, we're actually testing compounds in humans much earlier than would be the case in pharma. So I think this study of predicting that sensory data from the perception by human panelists demonstrates that the approaches could also be applied to predicting some of the very subjective human trial data in clinical trials using earlier experimental endpoints, and thereby prioritizing some of those very expensive clinical trial resources. What do you think about that, Samar?
Yeah, absolutely. So, this was an example of predicting the sensory properties in combining these properties with physicochemical properties. And in a similar way, the endpoints from clinical trials can be imputed using studies in animals or from in vitro screening, either target-based or cell-based. So a combination of these early experiments can help impute such subjective or like variable, let's say, clinical endpoints. Definitely, yeah.
Dmitry, one thing I always get asked is what next? Are you able to share a little bit about where IFF plans to take this project?
I think that's what would be the really interesting thing to do is try to apply similar ideas to predict the properties of fragrance mixtures. Because in the real world, all fragrance applications are mixtures. So there are mixtures of tens, and even in the case of fine fragrance, hundreds of ingredients. And it would be very difficult to predict that. And but with the help of methods like that, we're very, very optimistic that we could do it at some point.
That sounds fascinating. I hope we get an opportunity to work with you on that, or you'll at least keep us informed of how it's going. Well, thank you both again. And for those in the audience, if you're interested in reading the paper on this project, as Samar mentioned, the link is at the bottom of the slide. Now, I can already see that we have some questions, and we'll come to the Q&A shortly. But here's just another quick reminder to type your questions for Dmitry and Samar into the Q&A panel. But first, what I would like to do is take a minute to tell you all about how you can also access this technology for yourselves using Cerella, which is our platform that deploys Alchemite for deep learning or for active learning. So how you can access this technology at your organization. So just to give you a brief introduction to Cerella, and how it enables the active learning for compound optimization. Cerella seamlessly connects to your data repositories and brings the data in where you've got this sparse matrix of compounds on the vertical axis here and various experimental endpoints on the horizontal axis. It's automatically updating from the available data and preparing this data for model building. It applies cleaning and your organization's business rules, and it transforms the data for the best model performance so that Alchemite can build and update the models. And it goes through this process automatically updating the models in a sense of active learning as new data becomes available, so that you're always working with your results that are based on the latest data. And this removes that manual burden that's typical of QSAR modeling, of manually building and updating the models as projects progress. From here, the massive matrix of imputations is filled in and maintained, and this may easily contain thousands and millions of data points. And it's very easily accessed either seamlessly through the Stardrop application from Optibrium or through a RESTful API where it can be accessed in the same way from other platforms. If you'd like to learn more about Cerella and how it's working, we have a previous webinar that's available on our community website by following the link that's at the bottom of this slide. So now to the Q&A part of the presentation. And I'd like to start by thanking Dmitriy and Samar again for a fascinating presentation of this project and its unique challenges. I can see that we've received several questions, so let's get started with a few of them. The first one is for Samar. AI methods are known to work and make accurate predictions when large data sets are available. Can you talk a little bit about how Alchemite can show an advantage over QSAR on a data set of, say, about 1,000 compounds?
Yes, absolutely. So the conventional deep learning methods and AI methods can only start to show benefit over QSAR if large data sets are gathered in the magnitudes of thousands or 10,000s or even more. And so because of the unique method of leveraging both the structure-activity relationship and the imputation aspect of it. So there are two layers of information that are integrated in this case from both the structural or the compound descriptors and from the inter-assay correlations or inter-property correlations. So when these are combined, we can skip having lots of test examples or compound examples in order to generate accurate models, which shows the advantage of this technology over conventional deep learning methods and QSAR methods.
Great, thanks, Samar. Here's a question for Dmitry. Could you comment a little bit about how different fragrance molecules are from other chemicals?
Oh yeah, that's an excellent question because we didn't have time to talk about that in presentation. But volatile aromatic molecules are necessarily small molecules. They have molecular weight less than 300, and they basically represent this subdomain of the domain on volatile organic chemicals. They typically have only one, no more than one hydrogen bond donor and no more than 3 hydrogen bond acceptors. They're basically typical examples of fragrance ingredients. There are aldehydes, alcohols, esters, ethers, ketones. There are small molecules. So, and the main difference is actually in this hydrocarbon portion of the molecule that can be quite complex, has some kind of fused ring structures like in terpenes. It's very different from drug-like molecules, and it's actually quite amazing to see that Alchemite can work in this completely different, for this completely different chemotype. Quite amazing.
Absolutely. Yes, sound much more like what in pharma we would call lead-like compounds, but perhaps even more extreme than that. And Dmitry, another question for you. Would it be possible to apply the current Alchemite model to predict the properties of molecules from a specific odor class?
Oh yes, absolutely. What we built so far is I would call it a general purpose model because we used chemicals from different odor categories in our training set. But yes, but in principle, if we have enough data, enough chemicals from particular odor categories, for instance, animalic compounds, then we can actually definitely try to target this particular group and essentially use very similar protocol to build this targeted model. Yes.
Great, thank you. Well, I can see that we're pretty much up on the end of our hour. So if we didn't get to answer your question today, we will definitely reply to that through email. I wanted to just finish up by thanking both of our presenters again and encouraging you to register for our next event on the 16th of November, Harnessing Generative Chemistry, How Scientists Can Guide Compound Idea Generation, where we will be introducing Inspira, the newest member of our augmented chemistry platform. So thank you very much for joining us, and we hope to see you at another of our webinars.